It is awesome to have you here on MyFlora Friday. Matthew and we have been friends and acquaintances and colleagues now for, I'm going to go with about 4 years. I think where we met first was when we used to have conversations on Clubhouse every week on a show called The Dank Hour. Matthew would pop in and we would always invite him up on stage so that he could give his pearls of wisdom about pests, pathogens, and other issues in cannabis. And so, Matthew, you know you better than I know you. So, if you could go ahead and tell the people who you are, what you do, and how you got into cannabis.
Well, Matthew Gates, IPM specialist, and I've been doing it for about 17 or so years now. Sort of IPM support, not just in cannabis, but in other crops. I've got a lot of extensive experience in cut flowers and vegetable crops, various ones like tomatoes. And also when I lived in China for about 2 years in the early 2010s, I worked in tea for a little bit and also sorghum and a few other crops as well. At this point, I'm mostly working cannabis, but I live in California and I have a lot of experience in other countries and other agricultural minutiae. I would say that what's important about my perspective, if there is anything important, is that it focuses on a holistic, sort of multi-domain approach to plant health and pests. I think pests don't get enough credit. I'm not here trying to say that they're good or that we should be happy with them in our crops, but I think that sometimes we oversimplify nature, which I think is really it gives us vulnerabilities, unfortunately. If we don't look at things at the right capability level and threshold. These fungi, these insects, some of them are beneficial for us, but a lot of them, they were doing this before we were around, and they're incredibly capable at what they do. And if we don't give them that credit, then they'll adapt and they'll evolve all over us. And that's why I use this term, the Everswarm theory or Everswarm concept, for my sort of IPM threat analysis, so to speak. The Everswarm concept, I like to use this as a way to kind of simplify somewhat, but not to the point of like indolence. That, all these organisms, it's not just fungus gnat, it's not just two-spot spider mite, it's not just budworm. It's certain populations in different areas that might have been exposed to certain things. They go to the Galapagos Islands, so to speak, and they have all these different exposures to different situations. They evolve in those contexts, and you can be doing everything fine elsewhere, somewhere, oh, I only grow organic, whatever that means to you. I only grow a certain way, or I grow in this really nice region that's really nice for cannabis, that's good for its growth or its whatever. That's all great, but when something comes out that was never in your area and it has all these abilities that you've never dealt with, your plants have never dealt with, and they're novel, truly, you might have a problem. And it doesn't mean that you're a bad grower. It doesn't mean that you don't know how to grow or that your plants are bad. These two are oversimplifications that are just not useful and also not true. So, in order to be aware of this, you have to know that this is possible. These organisms can evolve very quickly, very easily, and your situation can change. Your sort of ecology can change. So, if you're kept abreast of this information, then you can make sort of strategic operational decisions. Sometimes there's no resources. We know information, but we don't have a solution for what we find out. That's unfortunate, especially in cannabis where we don't get a lot of infrastructural support. But this is also, in a lot of ways, how people tackle at a 30,000 foot level other crop problems. And there's definitely infrastructural support, hundreds of millions of dollars, for example, for the spotted lanternfly that's in the East Coast right now. You've probably heard of it. Did you get to
Indeed,
see
I
any
have.
when you were in New York?
Oh, well, I live in New Jersey,
Or New
so
Jersey?
I
Oh,
see
okay.
them. The instruction is to kill them when you see them.
Yeah.
And they're so invasive that people go out there with like an empty water bottle and just suck them into the water bottle by squeeze and it like sucks them in. And they come up with like, full water bottles of spotted lantern. They're so pretty though,
They are
but
pretty, I have to admit. They're
they're
pretty cool
so
looking.
they're so cool looking. They're so pretty. And I feel so bad when, but they also do a lot of damage, which is, they're invasive and they do a lot of damage, which are two big no-nos. So here in New Jersey, they're very cognizant of things like invasive species and preservation, conservation, recycling, solar power. It wouldn't seem like New Jersey is like that, but I'm in the South. Depends what you subscribe to. South New Jersey, Central New Jersey, the shore is really where I'm at. So yeah, I'm an ignorant Californian, so feel free to let me know how it is in Jersey. Have you seen the Jersey Shore?
I have. I'm familiar with it.
So I live about 20 minutes away from there.
Oh, okay, I see.
Yeah. It is called the Garden State for a reason. It is really, really beautiful. The sort of extension of New York City, like the Staten Island area, and then it goes into like New Jersey City. And that area is very industrial, which I think is where New Jersey gets its sort of bad name from, where, almost everybody in the United States is like, New Jersey, why would you move there? Oh my God. And I'm like, no, it's really nice, really pretty, lots of flora, lots of fauna. And, I moved out here to grow hemp in the forest, which was an amazing experience. And perfect segue into we were growing in hoop houses, which are not sealed. They're not, they're open to the elements, sort of open to pests coming in. We were growing aquaponically, which presents a whole nother set of problems or challenges, if you will. And I remember there were multiple times when I contacted you to say, either what the hell is this? What the hell is going on? And you were always really nice to help me. So I think maybe can you explain, I mean, you wrote, and you also wrote this chapter in this book, Integrated Pest Management Practices in Cannabis. So, like, I guess, when somebody comes to you and says, Hey, I've got this problem, can you help me diagnose? Can you help me figure out how to fix this problem that I have? Like, how do you go about doing that? What does that look like? I just need some words of wisdom.
First, I want to say that I usually tackle it the same way generally, unless I get a lot more information really quickly. Big part of it is just to understand the context. And sometimes I'll give talks on presentations or in audiences or on YouTube even. And, I'll mention like a certain product or a compound or a biocontrol or something. Invariably, because of the international nature of this thing, someone will be like, oh, I can't use it, or oh, this state doesn't use it, or this province doesn't use it, or I don't have access to it in my country. So it's all very contextual, and especially in cannabis, it moves state to state. So I have to find out what their issue is, what their growth setup is. And then to be honest, I oftentimes feel like from there, you have to find out what's just the best business decision for you to do if it's a commercial setting, right? I'm
Mhm.
a big fan of things like biocontrols, microbial, and otherwise larger macro insects, things like that. But like, if you don't have a good crop scouting regimen, I always talk about this, then it does leave you open to the ingress of insects and their population growing really well. A lot of pests are not really a huge issue if you've been able to construct your situation appropriately, if you have a lot of dexterity in what you're able to do, and maybe the people that you have, or the things that you have access to, or like if you're an indoor greenhouse, outdoor, like is it harder for you to like render some kind of environmental control? Like if you're getting a bunch of things coming in, if I tell you something in your indoor, it might be irrelevant. If I tell you something outdoor, it might be too expansive, it might not be relevant to your situation. And a lot of times I feel like people, and the last thing I'll say about it before we go into another question, maybe, or something more detailed from this, is that, like, I think oftentimes, like, people are trying to sell a solution. People are trying to, do something like this, like, oh, they go to their biocontrol guy. Of course, they're going to recommend biocontrols and things like this. But if you have someone on your team that's just multifaceted like that, then you can kind of say, okay, well, what's perhaps a new thing that's coming out on the horizon? What's something that is going to be just much more significant or sort of salient for your particular outcome. Because maybe it's not important for you to apply something. Maybe it makes more sense to just get rid of the plant in some cases. If
Mhm.
it's just one, or if it's a hot spot, maybe you just treat one area, you don't treat your expansive area. Maybe you're not in flower yet, so you can get away with doing that, and it can be less expensive. I love it when people try to address their solution by the nines and be super comprehensive. And in fact, the Everswarm theory I talk about is a big part of that is like any selection pressure you try to use, it's going to be a selection pressure. Even things you might think are harder to evolve a resistance towards, it's still there. And so in that way, it's good to be sort of multiplexed to account for all those variables. But you want to do that with the least options that you need so you can be most economically feasible.
Right. You don't want to just throw all the shit at the wall and hope something sticks. You want to do it with purpose and preferably with some sort of data or something to back up the decisions that you're making, right? Because
Absolutely.
and so, I contacted you a little while ago. I've been working with MyFlora DNA, which does multiple things, but right now we're focusing on pathogen testing. And, being a cannabis scientist, of course, you have to have pathogen testing, and some are worse than others. And you are a consultant that would come in and help not only diagnose, but then fix a problem because each pathogen it's not a one size fits all to get rid of stuff, as you were saying. Like, you may not need to treat a whole greenhouse or a whole room. There might just be an outbreak in one area, which you can get under control fairly well. Whereas if you have something like hop latent viroid that shares a whole water system, then that's another problem entirely, right? So, my idea was to come up with looking at the symptoms. So, what's wrong with your plant? My plants are turning yellow. All right. So, first thing you can do is there's a couple of pathogens that share that symptom. And so at MyFlora, we would test for three pathogens that result in yellowing symptoms. So then, if we test and it comes back positive, you have lettuce sclerosis virus. Great. Now what do we do? So in that case, they might have somebody, as you were saying, in the facility that knows how to deal with these issues, but they might not know how to deal with it, right? So then that would be, we can send them your information, be like, we suggest that you contact Matthew Gates. He can have a one-on-one with you, talk about your facility, what's going on, and then give you some sort of direction. But also, on the other hand, if they get a pathogen test and it comes back negative, you still have an issue, right? You sent your stuff in for testing for a reason. So you still need a consultant to help you figure out what's going on. Well, I think it's a good way to approach these issues. If you feel different, go ahead and talk about that. But like, that's more of like a holistic approach where you're just not trying to pinpoint like one thing. You're kind of going, there's something wrong. Let's figure it out.
Yeah, you have to, like, how many times have I gotten a question from somebody where it's like, what's wrong with my plant? And, it's like just the leaf is discolored. Maybe it's chlorotic, maybe there's lesions, maybe it's just kind of like yellowing, for whatever reason. And because a lot of pests and pathogen effects on the plant will mimic the same symptoms of nutrient lockout or some sort of like weird physiological effect, perhaps because that is what they're doing in some cases, you can't know just by visuals in a lot of cases. And like with your example, the sclerosis virus and similar other kinds of viruses, if they have an overt symptomology that doesn't change much and is very consistent, sometimes you don't necessarily need certain kinds of tests, right? So you do need the test, even if you don't need it for the identification, you need it to quantify, what your situation is. And then you can make choices after that. If you try to make choices before that, before you confirm that, you make it with not all the information. No one makes any choice with 100% of the information, of course, but you can have a lot more because it's possible in a lot of cases these insects, like with Lesch sclerosis virus, it's vectored by the silverleaf whitefly. And so, for example, let's say you get LCV back as positive, but you haven't found any silverleaf whitefly. Well, that's a real problem, right? That's really weird and concerning. You might not even have an answer necessarily. So you go crop scout, then you find some. Hopefully, but perhaps it doesn't go that way. What if you don't find them? Well, what's going on? You go
Mhm.
outside your property. Silverleaf whitefly. So your sprays or whatever management techniques, maybe you had biocontrols that go after them that keep the population low or basically curb it completely, like Swerskii, cucumeris, things like this. They go after the eggs of a lot of whiteflies and the larvae, but not so much the adults. But what I'm trying to say is you could have a situation where the vector actually gets killed before you notice it in your crop scouting. Maybe because your crop scouting could be a little bit more robust, or perhaps because the whatever controls you have are very effective. But the virus still gets transmitted, and so you still have an issue. But you wouldn't have noticed it, and you wouldn't have been able to know to what extent without, for example, testing and that kind of a thing.
So the flies got in on a Monday. They infected the plants on the Tuesday.
on the
They got
Monday
killed.
itself.
They got
Well,
killed on the Wednesday because that's when you spray for whatever, or that you do a treatment or something, and then you don't notice anything in your plants until Friday. You send it
or
in for
maybe
testing.
several days past.
Yeah.
Here's another monkey wrench. What if you happen to, by dint of fate, you just happen to have a plant that's resistant to virus X or whatever we're talking about, or more
Mm.
tolerant of it than the average one because we don't have data about this. And so what if your plants are actually kind of somehow doing better, and so that takes longer for you to find out?
Or bring in a plant that's infected with something but doesn't show any symptoms, and then gives it to something else, and now you've got a problem, but you brought it in without knowing. So testing and
Exactly.
quarantining things that you're bringing into your facility is important, as well as general hygiene and sanitation of the people that are coming into your facility, because bugs can kind of hide out like in hats or shirts or whatever. It's not just a matter of washing your hands, but you could be bringing in anything, right? On your shoes.
I worked with a grower one time in the LA area, and they had an indoor facility that was a retrofit of some building. They definitely did a lot of construction to anneal it into what they needed it. And so, but what I remember finding is they had a rice root aphid issue. And they were like, wow, I don't know what's going on. And the long story short is that their front door had like a inch, maybe three-fourths of an inch space gap where anything could go through. Not great biosecurity, but on top of that, they had recently gotten rid of ivy that was on their building, and
Mhm.
the like weedy barley that just grows up out of the sidewalks and on the all the like ruderal land in the urbanscape on their property, were basically carrying the rice root aphids. And when they had gotten rid of some of the ivy, they had jostled it quite a bit. I think it had agitated them and they started to move in. They had not come in since then, so they had a ticking time bomb this whole time, and then they did something, and then that's kind of a unique situation. At least that was the chain of events. And so you do have to put on your forensics cap and kind of make an assessment yourself. It's not a video game. No one's coming down and going like, "You figured it out." It's like, uh,
Level up.
right, exactly. So, I mean, that seems to be what had happened. Maybe the ivy had nothing to do with it, but I found it on both of those plants. So, seemingly that is what occurred, and they gave me that info. So, something like that can happen, and it's pretty common to my mind. And especially these viruses are very pernicious, and a lot of times you can't cure them conventionally, which makes dealing with them pretty difficult.
Mm.
So, you mostly focus on the vector and preventing the vector from getting into your space.
As somebody who is constantly diagnosing issues from multiple different angles, do you find yourself thinking like a plant, or thinking like a bug, or thinking like a pathogen of how they go about doing what they do so efficiently?
So thinking like an organism that's not a human is something you mostly only come across in maybe like really great sci-fi, but I think it's really useful for understanding your cultivation situation. So this is a slide that I've taken from a research report that's really cool. If you're into this kind of stuff, you should definitely check out the cited source at the bottom in green, this 2016 paper. And basically, like this shows how powdery mildew infects a cell and or how it fails to do so depending on the plant's response. But you can see that it is pretty robust, but it's happening even at the very top of the cell, the epidermis, right? So the powdery mildew creates a little what's called a penetration peg. Can you see my cursor?
Yes, I can.
Oh, perfect. This section's red part that says non-host resistance kind of shows two examples where powdery mildew will fail if your plant and all the other, gobbledygook, to use the technical term, that has to influence the phenotypic expression of the plant and the organism that's, both of those. So one example is it doesn't even get very far, the penetration peg fails, or physically, mechanically, or somehow like metabolically. There's a bunch of like enzymes that break down the cell wall and that kind of a thing. And so the plant will notice this effect. In fact, it will even notice when the hyphae are moving across from the spore across the epidermis. There's volatiles essentially and other sorts of compounds and chitin that breaks apart and gets sensed essentially. So if the plant's response is really comprehensive. If the signaling works out and the expression works out, then this can just stop here. And this papilla that gets formed no longer penetrates. It doesn't get to this point, which is this haustorium, which is like a
Mhm.
sac where the powdery mildew exudes a bunch of effectors, things that change the genetic expression of the plant at a cellular level, which is pretty fascinating. And there's also the sump pump from which it will take in the nutrients and sop up all the sugars. It even causes the genome to copy itself in the cells, which is kind of cool. Not for us as growers, but to know that it can manipulate the plant at such a fine, minute detail. So if this is not effective, what will still happen is the plant will produce these things called MVBs, these sort of vesicles, I believe. Yeah, is that the right term? Yeah, multivesicular bodies. And that will basically coat this haustorium and choke out the powdery mildew. That's another way powdery mildew can be affected. And this might be slower, so the powdery mildew might still be able to grow a little bit, but this will either hasten its demise or totally overmatch it. But if this doesn't happen, then the powdery mildew is probably going to be more effective. It'll produce tons of spores, which are what these conidiaphores are. These like spore producing bodies, these fruiting bodies. They're like mushrooms, but they're small. They're not mushroom forming fungi. And so to understand what powdery mildew has to do and to respect it appropriately as an opponent, let's say, you have to understand that like in the case of Golovinomyces siceracerum, the lettuce powdery mildew that we often deal with in cannabis, it basically like 60% of its genome is dedicated to manipulation of the plant host, and it's constantly evolving. We they used to call it a two-speed genome, where like parts of the genome would evolve at a certain rate. The virulence factors and virulence genes would evolve and mutate much more rapidly because they have a lot of transposable elements. You see, powdery mildews, right? They're mu much more complicated than just some white powder on your leaves. Anyways.
Just thinking about how the pathogen is going about its daily business, just trying to survive, and then the plant is going about its daily business, trying not to get damaged and killed or whatever. It's just really interesting to put yourself in something else's shoes. You talked about this idea of resistance versus tolerance. I had a conversation with Zamir Punja not too long ago, and we talked about this idea of plants being tolerant rather than resistant. Like they don't really not get infected, they get infected, but they're tolerant, or they do a better job of holding the line and not letting that pathogen get too far into it to where it's has to succumb to whatever fate happens.
Yeah, actually, there's a lot of discussion about, the terminology. So even when I talk about it, I'm passionate about it. I think you have to also concede that it's a fuzzy terminology. Many people know that like it's not just genotype, it's phenotype, and you can have different kinds of phenotypes, different kinds of environments. Obviously, that's still in play. A lot of people are talking about how, yeah, maybe the better terminology to use is like tolerance versus resistance, or there's earlier models, like my favorite quote, one of my favorite quotes is like, all models are wrong, some models are useful, right?
Yeah, I've heard you
Yeah.
say that before too.
Yeah, it's really true. And so like even if I'm dead sure that the diagram I just brought up is pretty accurate, there's obviously things that are not in the model that are being accounted for. So this slide talks about something that I think almost never gets talked about in the cultivation space, even though we just talked about this gene. And I'm sure you know better than me that gene tolerances or perhaps how different organisms interact. We people used to say it's gene for gene. That was when we first learned about genes. Like there's a gene that does this and there's an anti-gene or whatever, and they connect. But that's not how it works. It's very, very, very complicated a lot of times. But like for example, one of the things is like time and age of a plant and the maturity of the plant plays a big role. Obviously, at the inset, people can understand that a young plant is not as strong, maybe, or doesn't have the same capability as a larger plant, not just because the plant has more volume, it can take up more nutrients, but also because it's matured and, all these other genetic factors have been more expressed.
Knows how to deal with problems better.
Well, yeah, but also, well, yes, that is true. But the other thing that happens is that plants are masters of. We talk about how a plant thinks. Plants are masters of signaling, they're masters of detecting an issue and then also responding to it proportionally, which is the really important term here. And there's this concept called the growth defense paradigm. The idea is that plants have finite resources, even if they got microbial symbionts and all that stuff. First of all, those symbioses require resources to continue to work. And then secondarily, but you don't always have the same kind of output or input. And certain symbionts can even do different things or be more or less capable of the same process. So even plants have to make choices. But here we have an example of two different pathways represented by the jasmonic acid pathway and the salicylic acid pathway. There are different pests, and they achieve their objectives using different manipulation tactics. Powdery mildew was one of them. That's a salicylic acid pathway type thing. So plants get primed through this pathway, and then they're like, the next time a powdery mildew comes in, because it's probably going to happen, right? I just got it. It's probably the season. They're not consciously thinking this, but this is kind of how the process works, and it kind of makes sense. Maybe there's more fungi about, and so powdery mildew resistance genes, you could say genes kind of get upregulated. However, if you get something else like botrytis or fusarium, or you get caterpillars, for example, represented here, they prime a totally diametric response. It does totally different things. They contradict with each other a lot. So you can't, if you get both of these at the same time, like let's say necrotroph and a biotroph, so like botrytis and powdery mildew, or caterpillars and aphids here, then it's a bad day for the plant because it has to do two things. It has to look right and left at the same time, and it also has to grow. So we can do things like prime the plants. We can prime plants in a way that allows, with microbes and metabolites and certain nutrients, even, we can help plants prime in a certain direction, but we have to be cognizant that that can have a detrimental effect if we prime them in the wrong direction at the wrong time.
Priming plants is not like a vaccine. Like you're not priming it to
Right.
recognize something it needs to fight in the future. It's priming it.
But they don't have like memory T cells or anything.
Right.
It's their innate immune response. Constitutive defenses, you could say, that are already in play, and then there's pathways that get unblocked, essentially. Depends on the organism, I suppose. But like for botrytis, like for powdery mildew, you deny them the cells. You initiate the so-called hypersensitive response, and the cell lyses itself. Denied. Powdery mildew dies too when that happens. If it's the if the spore's like, I only got like 80% of my battery left, I better get a cell. I better get some sugars. And then that is the one cell it needed. Maybe there's lots of wax on the leaf, and it's like, oh man, I can't find a good cell. Oh man, I'm gonna die. And it finds that cell on its last legs, and it gets crumped. Then the powdery mildew is unsuccessful, and we are.
And we win. Does that priming come from exposure where the plant has a low level of infection, is successful at clearing whatever is going on, and then the next time it has a faster response because it already recognizes that thing?
In nature, that's usually what happens. Like it gets exposed, but it can also be exposed to non-pathogenic fungi. And it's like even mycorrhizae, for example, and I have a slide about that too. But like mycorrhizae, just the exposure to them or even the association with them will cause the immune response to the immune system of the plant, if it's amenable to mycorrhizae in the first place. Then it also triggers other fungal sort of like blocking measures or like barricades you could think of metaphorically. Because when the plant lets the fungus into its body, it's actually like a bouncer kind of. It's like you can come into this room, we let you set up here. You can't, we're not going to let you grow out of control. And I think a lot of people don't realize that it's this very like parlay interaction. And it's not like that the plant totally trusts because you can get cases. That's how powdery mildew become a pathogen. It used to be it evolved from like beneficial fungi like long, long time ago that would break down plant materials and things like that. But it learned how to use that secret code to talk with the plant to then rob it of its resources.
And now, does this happen differently in different regions because there's different natural or native pressures in different regions around the world, right? So if you have a plant, like let's say you, I don't know, bring over a plant from Hawaii, let's say, and you want to grow this plant, is it going to have a hard time because it's naturally primed, if that's the right word, for things that are potential pressures in Hawaii versus, let's say we want to grow this Hawaiian plant in Colorado. Is is that a big deal or is like,
It
you know,
could be like, this is just as an idea to make the scenario. Is it like, did you just take a small seed and you grew it in a new place, or is it already fully grown for your
like a
scenario?
plant, like a cutting, maybe like a clone. So
Yeah.
it's already like an adapted plant. It's been under pressure or it's had selective pressures. Maybe it is a locally adapted variety and maybe it's a seed, but let's go with the cutting.
Okay, yeah. So I would say it could be really good. Maybe it does better because there's this concept, this hypothesis called the enemy space hypothesis, I think it's called, and there's a few others that try to seek to explain how, as the name implies, if you take an organism out of an environment, put it in a different environment, maybe one that's conducive because of the sun or the abiotic factors, very common in California, but also because there's no bad guys to attack it potentially, or at least not the right ones, or ones that aren't as effective. Humans have kind of upset this a lot. You bring it to the desert and you don't have any humidity, maybe it doesn't do so hot, potentially.
So you mentioned mycorrhizae. I am not super familiar. Like I know kind of what it is, but I'm not like and I'm very far removed from an expert at this. Like I know that mycorrhizae are important for interactions and I don't know, health, not health. I don't really know. Like you know what? I'll shut up. You go ahead and talk about mycorrhizae because you know about it.
Yeah, like mycorrhizae is an interesting topic because like I kind of feel like the bad guy sometimes, but I'm okay with that. I'm okay with being the guy, just like you, it sounds like, who's like, hey, maybe this terminology or this concept is not totally true, but maybe there's hints of truth in it, and we should explore it. We should just confirm one way or the other for certain topics, and also that kind of thing. So, not to appropriate what you said or anything, but I do think it's in the same kind of pursuit, which is like, sometimes things we normally call good are not good in some cases. What a concept. So here's what I mean. I love to use this example. This is from a paper that talks about arbuscular mycorrhizal symbiosis. So these are fungi. For those who aren't familiar with them, they basically are specific kinds of fungi that colonize plants. Old terminology used to say infect them, but I like to use the word colonize better or somehow associate with them because sometimes some mycorrhizae, like, they only get onto the sort of outside cortical layer of the roots. Others, they go right into the cells, they invaginate the cells, and they're right up into the cellular space, and they're exchanging nutrients and water and sugars and minerals like right up there. So there's different kinds of relationships that have evolved over time. Some plants actively do not interact with mycorrhizae. Some are pretty famous. Others are just very inexplicable. For whatever reason, they lost the traits to be able to do the Nike and Mike factors and these sort of things. On this diagram, though, I have examples of this particular fungus in the early stage of a plant and the late stage of the plant, where they had effects on viral infection. So there's cases where the mycorrhizae limited virus infection. It did things that would typically limit its ability to move across the plant. Part of that's by stimulating certain immune responses. We talked about salicylic acid. They produce things like ROS, certain R proteins, and other factors like that. But they could also enhance virus infection. So things like abscisic acid, not only do they drop what are called callose deposits into the phloem, basically big woody chunks that literally block off movement and transfer of fluid, The abscisic acid will disrupt this, or will disrupt salicylic acid as well. It's because they're kind of contradictory. So you get a multitude of effects, and it's not enough, I think. It strays from being informative and strays into marketing when people just say mycorrhizae good all the time.
Mm.
Maybe it's usually good, maybe it's 99% of the time good, but maybe sometimes you don't want the plant to expend a bunch of resources. You already got that priming figured out. You went to my videos on YouTube or something. So my major point here is that as you get other organisms like these aphids are the most common vectors of plant viruses, bar none, basically. And so they also suppress or otherwise disrupt the immune response when they're sucking down the plant sap. Their stylet, they call it, is so fine that it can go into an individual plant cell, penetrate it, do a little taste test, a little lick, and it doesn't even rupture the stability of the plant cell. So, and that's how the viruses get through all of your defenses, all of your immune things, all of those cool microbes you spend a lot of money on, right into the plant cell, right where it can do what it wants to do. So, yeah, so it's not just the virus, it's the virus on the aphid, on the air.
So aphids, you say, are the by far the biggest vectors of plant diseases. So aphids are a big deal, right? And but one of the biological controls for aphids is ladybugs, right? That's the one that everybody talks about.
That's
But
what a lot of people do talk about.
in my experience, it seems that if you give a ladybug a chance to leave, they will.
Yes.
But they don't like to stick around even if there is a food source. And ladybugs don't really eat anything else. So when you purchase ladybugs as a biological control, is that really a good um use of your money if they're just going to leave at the first chance they get and the only thing they eat is aphids, or am I completely off base?
Well, there's a bunch of lady beetles out there. There's some that eat spider mites only. There's some that many eat aphids. Some even eat powdery mildew, if you can believe it. And are being investigated for powdery mildew control. Although the research I'm familiar with kind of shows that the larvae, like they'll spread the spores as they eat them, so it's not really ideal. But I generally don't advise people to use lady beetles for a few reasons. One of the reasons is that a lot of times they are harvested destructively to the environment, and the reason why they often fly away when you release them is because they're harvested cold. If they're not already like the harlequin lady beetle, Harmonia axaridis, which is invasive to the USA, it interferes with other native lady beetles, the ones I was just talking about that are harvested, and they rarely give them diseases of some kinds too, but like they're basically a menace. They'll bite you, they'll excrete a bunch of yellow, very smelly, very acrid substances that stain as well. They kind of get into your houses looking for places to hide during
And
winter and autumn.
are those the ones that are they tend to swarm together?
Well, a
Whereas,
lot of lady beetles will swarm together for when they like basically aestivate or hibernate kind of. But yeah, like stink bugs do it too. A lot of other insects will do it as well. And so yeah, I don't like them. I don't like those ones in particular for all the reasons I mentioned in North America, but also even the other ones that are native to here, the way it works is they huddle down, they live out the storm, the winter, and then they come out and they fly because there's
Mhm.
probably nothing where they are because it's all barren, right? When it warms up. So their first thing they're going to do is go find food and or mates or both. And so when you get them, they're harvested when it's cold. You go to open up the can because they're kept cold so they store for a long time. And maybe some of
Mhm.
them are dead, maybe some of them are old. But on top of that, the ones that aren't are going to behaviorally are not going to want to stay around for that reason.
So I don't know if I missed it, but are they worth spending the money on? If I mean,
Nah,
are they meh?
I don't think so because they'll fly away.
Yeah.
In those cases, or they'll fly into your wind blades. I guess that's the implication I'm making. I think that there's almost always better solutions. Lacewing larvae are really good, but they're also expensive, I feel like. So, in my opinion, And for the cannabis in particular, you mostly get two kinds of aphids that are important. There are
Mm,
other aphids out there that I've seen, bean aphid, maybe some peach aphid, which are really common generalists. Never been a problem for anyone I know, and I don't
mm,
expect them to be. But the ones that have been are cannabis aphid and rice root aphid. And rice root aphid is a big problem because it lives hypodulally in the substrate. And none
mm.
of these biocontrols are going to get there unless it's like a fungus like Beauveria bassiana. So that's a solution, for example, a biocontrol you could use, but I wouldn't spend a bunch of money on like insect biocontrols for them. But cannabis aphid only exists in the foliage. So you might have a lot more success. Indeed, I've worked with Copper on a field basis. They were trying to develop a new lacewing product with eggs and larvae and which combinations made the most sense and cut flowers. And they found that basically the lacewing larvae are really effective when you have a huge hotspot. If you only have them like a little bit of pressure here, a little bit of pressure here, whatever, it's not really worth it. They have to go searching a lot. It's not as efficient. If you're a small home grow and you've just got a tent, that's different. But if you're
Mm.
at scale where it matters and these sorts of costs start to pile up, I think that would be a better solution or to not use them or find an alternative.
Yeah, we would get like a ton of ladybugs and lacewings and let them out, and then they'd only stick around for a little while, and I never really saw the point. Something else that we did that I thought was kind of cool, and I don't know how effective it is, so I'm going to ask you, spreading nematodes around the property, and yeah, and in the grow, we had living soil.
I like nematodes. I think that they're actually my favorite control for fungus gnats and shore flies and these sorts of flies that the larvae they love like propagation. They love like humid, moist conditions. You got like a gutter with a bunch of algae, they'll really love you for that. But the nematodes will make a giant meal out of them. And you almost always get a really good price, I think, for a lot of the big name companies that supply them, and you can store them for a long time. Not forever, but you can store them for a pretty long time. You don't have to use all of them at once. You should use all in a packet that you get them in or whatever, however that is done. But like basically you can buy a bunch of them and have them in reserve in like a fridge, which is really, really useful.
Like buy a case and then open one container, spread it around, and then a couple
100%.
months. Yeah. Okay.
Yeah, and then you can just have those kind of on repeat if you're like, wow, if you don't have any other way to solve the problem extrinsically, like if you keep getting fungus gnats, maybe there's a reason for it. But if for whatever reason you're just open air propagation or whatever, everyone's different, people
Yeah.
trying to make it happen. But there's options then. So a situation that might feel untenable suddenly isn't.
Gotcha. And so what is your favorite? I mean, I guess you kind of already said this, but it kind of depends on your sitch, right?
Yes.
Of what you would call your favorite, but I'm sure you have your favorites for, you know,
For what?
give us your top three favorites.
My favorite biocontrols are probably predatory mites in general. A lot of them I like them because they're versatile. I like them because several of them, the type 3 generalists, they call them, like Cucumeris or Swirskii, they feed on things like thrips, like whiteflies. They feed on different life stages, egg stages, larval stages. They'll even harass the adult stages, which is good. So, and they're oftentimes really easy to access. They're accessible in a lot of places. There's a lot of companies that make them. There's other predatory mites that are a little bit more specific, like Californicus. But these Cucumis and Swirsky, they can also feed on pollen when you don't have any pests, which is great. Or you could have banker plants that produce pollen and they can live and sort of exist on them like a sort of living garrison. And I've been effective with people using those as like a augmented biocontrol, especially in greenhouses. So I like them for that reason. There's a lot of ways you can use them creatively and smartly if you have the right setup.
Okay. All right. And so I was just thinking about our days on Clubhouse and then into the dank hour that was on Future Cannabis Project. And our friend Ashley, do you remember Ashley? She worked in a cultivation facility. Well, she still works in cultivation, but she would utilize these packs. And you probably know exactly what I'm talking about, but I don't know how to describe it because I've never seen them. But I remember her describing them, and they were full of, I don't know if it was eggs or what, but they were like timed control where like it would slowly release the biocontrol
Oh
over
yeah.
time, and she would tie them to her plants and they would last for like a month instead of dumping a whole bunch of ladybugs or whatever. So
Yeah.
instead of doing that all at once, because they're live animals, so you can't leave them in the package, these would slowly release over time and they would kind of crawl out of the packet when they emerged.
Yeah, the bulk release has its place, and what they call like a slow release or an augmented release, augmentative control. One of them is bulk, giant army, hotspot, dead. The sachets and other things like that are kind of meant to be used like long term. Different products will have different ranges and whatever, but generally it'll be like 1 to 3 weeks, usually 2 to 3 weeks of like control of like you'll basically have the predatory mite at a certain population in your crop if you put enough sachets per meter squared or whatever. So I find them super effective. It's probably my favorite biocontrol like success story is using them. I'm very successful with them because they're a product that's very refined. It's been many decades that this kind of use of the predatory mice has been not only envisioned but then used and honed. It's no surprise to me that's so effective against the pests that they're for, at least.
Gotcha. Okay, I've always been really interested in those, but I've never had the opportunity to use them. And then, the sticky paper that people use to identify what bugs do I have in here. You can use that, the yellow or the blue stickies to catch the bugs to figure out how to attack your problem.
It's like, and there's automatic versions of this now, and AI vision versions of this now, where you can take the cards. I told someone recently where, I used to work with a cut flower grower where they had a guy who would go out and apply, he would take so much time, not because he's bad at what he does, but because the process wasn't very efficient, right? He brings
Hmm.
out all the sticky cards, put them all in each greenhouse. Each house is like 4 or 5 rows, and then he would put them all out. At the
Hmm.
end of the week, he would collect them all. He would go into his little office and he would look at all of them by hand and write how many were in each square for each card by hand for each greenhouse on paper. Then he would collect all the little papers and he'd go to the computer and he would then type in them all there. And then we would use that information to track. In this case, it was mostly leaf miner flies, which I sometimes see in cannabis, and we would use diglyphus wasps that would parasitize them. And you'd see the populations of both, they would go like this and they would follow. We would
Hmm.
use it also in combination with temperature, or at least when I came, that's what we ended up doing. We truncated the whole population because we could tell in the summer it would go all the way up, and we could tell there was going to be a big crash. It's because if the adults are getting on the sticky cards and there's a big amount of them, then we know in the next few weeks there's going to be a ton of larvae that they've inserted into the leaves. So then that's when you apply the wasps that will parasitize all the larvae, and then you know you've succeeded because there's not a big intake the next week.
Cool. So on a really small scale, like the how wolf and deer population kind of follow each other. Like there'll be
Exactly.
more deer one year, and then they'll get, then the wolf population will boom, and then the deer won't be there anymore, so they won't have much to eat. So then the wolf population will decline. Or you could do it with anything, like,
Yeah. No, but
rabbits
you're
and
right. That's exactly it. That's exactly it. So, you got to understand, you got to have that data. Otherwise, you're just blind firing. You don't know what's going on.
yeah, yeah. That's cool. I never even thought of that because I don't think on this, m minute scale of these tiny little things that, affect our plants in multiple different ways.
Yeah, I gotta say, did the genomics person tell me that I'm the one looking at things at a small scale? I appreciate it.
I mean, there's small scale and then there's
Really
small
small
scale.
scale.
Yeah. Oh, what was I gonna ask you next? Oh, I want to talk about powdery, is it the powdery mildew? So, I talked about Zamir and I having that conversation about tolerance and resistance. And I know that when I was doing the cannabis book club with Corey and Molly and Casey, I don't want to forget about Casey. We read a paper about powdery mildew and tolerance. It was really about how to treat powdery mildew and which treatments are better versus others and whatnot. But they also looked at tolerance or resistance, whichever one you want to use, for different strains. Like, are there different strains that are more tolerant than others to certain diseases? Like, we think about, hop latent viroid, and is it possible to develop strains that are more resistant to hop latent viroid than others, right? So same thing with powdery mildew, same thing with other things. So I know that there is some cool research out there on powdery mildew and probably other pathogens as well, but can you talk about that?
Yeah, I like this topic a lot. I think that just to mention the points we had said before, some of the cool terminologies that have been used in the past are like antibiosis, anti-zoonosis, which are like terms that kind of mean like what they're trying to describe with these words is what what does the plant do for itself in response, and what does the plant do to the target, the pest or whatever it is. And so, like, some kinds of like resistance are going to disrupt things going on in the bug's body, let's say. Other ones are going to be things that the plant does to itself. And you can see how it gets fuzzy because this is sort of like
Mhm.
kind of meshes together, right?
Yeah.
As the way genes and interactions do. So that's why it's the old terminology and sort of current terminology. Now people are looking at different ways to kind of describe it because tolerance, the thing that makes tolerance attractive as a concept is that the plants can take it, they can tank the damage, and then they can just keep going. And I've seen this in cannabis, I've seen this in other plants where certain varieties, for whatever reason, like let's say you get aphids, I've seen cases where plants will have like hundreds of root aphids on the foliage, on the roots. The plant's growing like gangbusters. It's no problem. Plant just keeps going. Obviously, you don't want the bugs on the plant, right? It's amazing how the plant just kept going. You just keep feeding it nutrients, keep giving it what it needs, and for whatever reason, it's just fine. Other plants you have the same amount or less, and you start to see chlorosis, you start to have stunted growth, maybe the plants are really young. So this can happen, right? And so I think tolerance is a really good way to put it because some plants they might get damaged, but it doesn't necessarily highly affect the yield or the quality of the product or whatever. And of course, you
Yeah.
still want to treat it because it's not then spending resources against it. But for whatever reason the plant can shunt itself off and keep going in whatever way that is. And so this research report's really cool because they basically looked at powdery mildew over they had different varieties, different cultivars, different populations of cannabis plants, however we want to put it, right? And that's over here on this axis. And then this here, this AUDPC, this is like the disease amount, essentially. The amount of disease they quantified without
Mm-hmm.
getting too much detail. And I've noted some of these, right? Like white CBG over here, higher. So this was higher susceptibility. This is PM susceptibility range. So high to low. And you can notice some hemp populations, right?
Yep.
Futura,
Those all look
Vibroor,
like they're all hemp.
Bayox. Yeah, it's all hemp actually, I believe. Yeah, you're right.
Yeah. And what I'm
Um,
noticing here too is that it seems to be that CBG came on just fairly recently to the market. Cherry wine was like one of the first CBD types that we started growing. And then Bayox and Auto 2 are even older than cherry wine. And then Futura and Fiber, those ones are like even older hemp cultivars that we've been using for a long time for fiber and stuff like that. So I'm wondering if it has to do with selection, whether it be us or nature, over time, because this is showing to me not only a susceptibility, but also a timeline as well. That we've got the new things that seem to be more susceptible than the older thing, the things that have seen some shit.
I've heard some people balk at the introgression, if that's the right terminology, of hemp genetics into, breeding stock, essentially. People saying that they don't like the idea, they think it's bad, that's going to make the plants bad, whatever. I don't know how true that is. I think that depending on how in the weeds you want to get, it's totally untrue because where do you think hemp came from? Where do other plants come from? And I think there's a lot of influence, especially in North America. I've read that there's a lot of Chinese and European hemp fiber and fiber plants in particular that were grown and that are part of a lot of landrace populations that are in now North America as well. So like there and then they've had like 200 plus years, 300 years now of like adaptation to the area. And I've read research about specific genes and profiles being unique to these land races, which I think is kind of neat. And then this is 2023 and this is 2024. You know, does do any of these names pop out at you as well, like photo CBD or anything like that?
So I don't know what photo CBD is, but yeah, cherry wine again. Carmagnola is I think that's an older, like I want to say it's
I
still
feel like so.
flower.
I don't remember either
I
though.
think it's still a flower strain though. I'm almost positive that it's a CBD flower. ACDC is also a CBD flower, CBD heavy. So is Otto. And then Fibroir 79. I recognize the name, but I don't know what its purpose is. But yeah, again, I think we're seeing also a timeline of newest to oldest. And for people who are, against putting in hemp or introgressing hemp type traits into our, smokable cannabis. Where do you think our autoflowers came from? They came from an autoflowering variety of hemp, and bringing in some of those genes allowed us to create autoflowers. And there's a lot of people out there that don't like autoflowers either. They're photo all the way. But autoflowers are incredibly useful for people who are short on time, short on space, you know? And there are some really, really great autoflowers that people have developed that are good yielders now that we've been working with these plants for a while.
I don't knock it necessarily, and I think that there's probably some hardiness traits that I believe are probably there and have yet to be fully unlocked. In some cases, might even be gifts from their naturalization into North American ditchweed phenotypes and that kind of thing. But yeah, resistance is absolutely a spectrum, as you can see, and so you cannot assume necessarily one or another. When I think of genetic resistance, I'm almost always thinking about proteins and enzymes and the molecular missiles that both groups kind of fire at each other and produce either on hand or in reaction to a threat. But that's not all of it, and disease resistance can be affected by things as simple as whether the plant's a late flower or not. And that was one of the things the study pointed out, which was really fascinating to me because three of these resistance populations were also late flowering populations. Some of the most susceptible were not. So they derived this idea that late flowering, which they define here as past September, some of these are flowering around July, August, but they're saying these ones that took a lot longer until mid to late September, maybe this could be true. Also, I think it's also true for like budworms. Budworms, they only colonize the floral tissue for the most part. So if your plants flower too late for them or the reverse, too early for them in your region, then it's possible that you might sort of eschew them, which would be great because I have this video about budworms that I posted on my YouTube channel, Zenthanol, recently. And it's a deep dive into very technical information about the probably the worst pest in the US and probably globe. You have all of these different regions where different resistance genes have been found. The American ones I have here in their location, more
Mm.
or less, but we've also got populations from, trade partners, really important people like the People's Republic of China and the in Brazil, where a lot of these resistances are happening because of exposure to certain toxins or the use of certain biocontrols and just having a massive scale. Brazil is extremely agriculturally competent, and the PRC has been doing it for a very long time as well. And so we even see resistances here to like, I'll name some of these. On the Chinese side, we've got BT resistance genes. BT is Bacillus thuringiensis. It's a bacteria. People apply it. It's considered very safe and effective. 11 resistance genes to this biocontrol. this Tret 1 and PGRP allow it to survive, as they found in Xinjiang, which it gets very high and very cold. Their pupae are able to survive negative 20 degrees Celsius in the soil, which is fascinating. And it would make these places here above like the 42nd parallel in the North America area where we count on the weather to kill them for springtime to not have them, they will be able to survive it and they'll be able to colonize this area instead of moving 700 plus or 2,000 kilometers from like Mexico and the southern areas to get warmer quicker, if that makes sense. So there's a global sort of strategic mindset that you could use to make your informed decisions. And then the last ones I'll say here are actually T-span might have been one of those cold resistance genes too. But yeah, here even resistance to Enterobacter, or I'm sorry, excuse me, because and here's the crazy part. The good microbes on your plants, some of them anyways, are also good for the pests. And it's been shown in research that budworms will co-opt a lot of core microbiome bacteria and fungi even from their hosts and put them in their gut, and then they allow them to colonize the plant better. They'll produce toxins that affect the other bacteria and other microbes, or they will break down the cellulose and other things that the caterpillars eat. And Enterobacter and Enterococcus, very famous, very popular, very commonly applied. These are two of the biggest ones.
And just for people out there who don't know about budworm, so you've got pictures of moths up here. I assume it's the same moth. That
Yeah.
moth has the baby or has the egg, and then that grows into a caterpillar or
Yeah.
whatever, which then does what?
Which then just bores into your bud. And it's so small. It's very, very tiny. I have a video that will go into more detail here, but like, yeah, they start as like a 1 to 2 millimeter white egg on your flower. Very, good luck finding it. And then in a 1 or 2 days, the larva comes out. It's very tiny too. It maybe stays outside for a few hours eating the leaves, and then it bores into your flower. Again, good luck finding it. And then it will start tunneling through your bud and it will cause infection, lots of wounds. It'll poop in your bud. That will also cause secondary infections. And what's worse is that even if you were to control it, even if you were to snap your fingers, right, and kill all the caterpillars in your bud, now you have budworm cadavers in your flower. So it's kind of a paradox. You either get it early, don't get it at all, or get a lot of problems.
Okay, so it's not necessarily that they're carrying problematic diseases or create toxins that would be harmful to ingest
No, no,
or
not in that way. They're mostly just a huge, huge nuisance, and they're really good at surviving the things we use against them.
and so really it's just destroying your crop.
Exactly. Total crop loss is very possible. I mean, we get like probably billions of these moths, especially in the Corn Belt, sort of area. So like Iowa, Nebraska. I'm a Californian. I have a slide that has more of these states. I don't want to like not name anyone, so I'm gonna leave it there. But there's
Call,
there's
you got to call everybody out.
yeah, exactly. Well, no, I mean they have lots of experience, and I've had people go. I'm going to go to Missouri in April, and in Columbia, Missouri, and I'm going to talk about this too. They tell me about it all the time. We get budworms. I don't know what to do. Honestly, a lot of times, if you want a solution, don't even deal with any of these resistances. Just get, if you have to. It's easier to do, obviously, but like a mesh screen or like some kind of walls, four of them, maybe a top that allows the budworms to not get it. If they don't make contact with your flower, the caterpillars aren't like other caterpillars that will like army worms will like move across the ground, get to another plant. They're mostly isolated. They'll travel across the crop if they exhaust the flower material. But if the moths never deposit their eggs on your plant, you basically don't have to worry about it. So that's why I think it's really essential in a lot of cases for people to build these sort of external defenses. Unfortunately, I know that is sometimes anathema to the goals and desires of the grower. Maybe they want sun grown or certain things, but maybe your region was good for some time, and maybe you'll have to find alternatives if you don't want to go a certain route. You know what I'm saying? So it also depends on what you're trying to do as a grower. That's where the rubber meets the road. It's easy for me to just say, use this, don't use this. But if it doesn't mesh with your plan, then it might not be useful for you.
So let me just get this straight. So these genes up here that you've got on the screen are resistance genes for the moth.
Yes.
So, for example, the Chinese ones that they've got a cold resistance gene. So that means that they are resistant to the cold that would normally kill them. Or a BT resistant gene, they're resistant to a pathogen that would normally kill them. So things that you could potentially think about using in protecting your crops from these bugs may not work on some of these because they have these resistance genes. Is that right?
100%. And like look this 2016 one here, for example, the BT resistance genes Cry1A, Cry2A. It only took 20 years for BT, like GM crops with BT in them, right?
Mm-hmm.
So those BT proteins, Cry1 and Cry2 and other ones that were used in like soybeans, it only took 20 years from the first instance of them being used to now that there's tons of BT resistances because of that selection pressure. So now what people do is they have crops that don't have the BT resistance or the BT protein crystals in the plants or however they achieve that objective. And then they have plants that have none. So that way there's caterpillars that get the resistance exposure and those that don't, and they mix and they're not as effective. They don't get as much resistance progeny from it. So, there's all this planning that's going on. And we as growers in the cannabis space, not only do we not get access to data and infrastructural research like we could in other crops, but also a lot of people are just not aware. It might even be their first time growing, or it's the only plant they've ever grown before, only crop they've ever grown before. Nothing wrong with that. It's my favorite crop too. But like, yeah, like there's all these things happening to be aware of, and they're definitely going to affect you. They're coming to a place near you.
And we don't get this data and analysis like we do in other crops, as you just said, because cannabis is still federally illegal, and we don't have the institutions that can actually study this like they do with other crops. So our ag schools and things like that. And that's why cannabis research is so far behind. And, everybody says, oh, but there's so much research on cannabis already. And it's like, yeah, but it's mostly in terms of what addiction, number one, like
Yeah.
bad devil's lettuce, don't smoke it, it's gonna it's gonna give you schizophrenia and all that. Like that's where a lot of the research has gone, is trying to prove that cannabis is bad, which none of it has really. Specific molecules that the cannabis plant makes and what that does in terms of receptors and the ECS and all that. In terms of crop science, though, it's very much been limited to hemp types because since 2014 we've been able to do research on hemp. But and we just kind of, because it's all cannabis sativa, we're like, okay, well, if it applies to hemp, then we can kind of extrapolate that out and say it would also apply to THC cannabis, type 1 cannabis, type 2 cannabis. So we were talking about resistance, right? Or tolerance to powdery mildew. I also think that, and this is just a theory, I have nothing to back this up, but it's kind of like a finite amount of energy, right? And so if you are putting all your energy towards something like making THC, for example, there's a trade-off there. Like you're not paying attention or don't have enough resources or energy to be fighting off pathogens. So I think that the selection for higher and higher and higher THC or higher and higher and higher CBD, we're kind of tipping the scale so that the plants don't have enough energy to fight when they need to.
Well, how about this? I mean, that's absolutely right, in my opinion. I mean, it's true. They say that maybe CBDA and THCA and some other products of certain other cannabinoids might have had. And research has shown that if a plant makes a new compound, it's like 90% of the chance it's going to be because it kills something that is going to have a problem for the either the individual plant or its ability to reproduce, right? So I don't deny that. But let's look at like, do you know what apples used to look like? Right? Do you know what teosinte used to look like? Like we make the apples fleshy and juicy and full of sugar, which they weren't like. And then we get gobsmacked when the pests are able to feed on them easier. Or like we get
They
rid of
want
the.
that juicy sweet apple as well.
Yes, yes, exactly. People think, no, that's human food. Bugs don't eat human food. I'm like, what are you talking about? That's so bizarrely not logical and not like ecologically read. But your depiction is really good, but also let's look at flower size. Let's look at yield. Let's look at, maybe because when you see the ditchweed phenotype I was just talking about in the Midwest, are they chunky buds? No. No, they're not really, because does it need that to survive? No, in fact, it's a liability potentially. Maybe you get more reproduction, maybe you get more seed production, but if the ratio of survival is too low, it won't matter, right? And so I think that we even if we're trying to grow in a certain way that is meant to mimic nature or close loops or be regenerative in this sort of way. I mean, that is a noble cause that I support, but it's also not really the same as saying that it's like nature because it's not really domestication and all these other things. I mean, we might have even been selectively pressuring cannabis before we were growing it in an agricultural way by just consuming the products and leaving the seeds out. They say that this is how certain other plants have also been, like hazelnuts are like this. In fact, hazelnuts, if I'm remembering right, a lot of them are not even domesticated because they outcross so well. So
Mm,
people will grow these orchards of hazelnuts that are like 300 years old or whatever, the population. And it's just they never had to like domesticate them. They're just kind of there,
mm,
if that makes sense. So there's still varieties that people grow, but like you'll also get situations just like in cannabis where people are like, oh, you can tell this is Tom Bull, big nut, the one that people want for all the confections and stuff. You like go to do genetic sequencing, and you're like, that's not Tom Bull, that's not Tom Bull. You were only characterizing it because the nuts were large,
hmm
not
Yeah.
because. And so similarly people be like, oh it smells like this or that, and it's like, well, look at the genetics, it's not this or that, so you were
hmm
wrong. It can happen with the Mark
Yeah.
1 nose. You can
Yeah.
make mistakes.
There is a distinct possibility that some of this ditchweed or these landrace strains have a higher chemodiverse than what we currently have in our cultivation or, these modern cultivars because they're using those molecules for defenses, not for our pleasure, right? So they might
Sense.
be producing things that are not necessarily desirable to us. And so, like skunk number one, David Watson bred out the skunk smell. Even though it's called skunk number one, he bred that out because he didn't personally find it pleasing. And so, people are on the search for the skunk smell. And it's like, well, it's largely been bred out. Is there some land races out there that might still have that as a defense because maybe some bugs find it nasty or maybe some animals, some herbivores, find that nasty. So that's like super
Yeah.
interesting to me because we're breeding out the diversity, literally, like not just the chemo diversity but just diversity in general by choosing and picking a few select traits. We're kind of emptying the toolbox that the plant has, right? And picking our favorite tools, and it's all it's got left to play with. Like
There's
cannabis is
even
it's very much a monoculture at this point.
I mean, in some regards, yeah, absolutely, right. And like specifically just the fact that people are growing it, they're breeding it, they're putting an intention in there. Nothing wrong with that. But yeah, I mean, you can't, if you go the left direction, you're not going the right direction. It's black is not white, like it's that's all there is to it to have that happen.
Hmm
Red is not blue. So
Yeah, right. We covered a lot of ground here today, I feel like, and it's been super interesting. You have a YouTube channel where you do what? Who can benefit from your channel and what can we see there?
I think that the thing that I do most on that channel is talk about concepts that I can't talk about anywhere else that have to do with plant health and pests. And like I said earlier, if you want to deep dive the pests that you're going to deal with, especially in cannabis, but some other crops too, because a lot of the pests in cannabis are pests of other crops. So very common generalists, spider mites, deal with the same spider mites, the same fungus gnats, the same budworms that we just talked about. So if you want to learn about those pests and understand their abilities, but also their weaknesses, then you should check out those videos. Look up Zenthanol Consulting or Zenthanol on YouTube, and you can look up the Pest Primer video series where I go over several pests in cannabis and other crops that assail us and what we can do about them.
And so your channel is really a great source of information just to kind of get yourself familiar with various past pathogens, your advice when you come across these things, what to look for. And then you have the consulting service to back that up. So if somebody does, have an oh shit moment, you have that consulting service. And where can people find you then? Is it, I'm assuming it's all over your channel and you're like, call me at this number or email me at this email address. But I know you're also very active on LinkedIn too. And that's a great place for people to have conversations. But yeah, where can they find
That
you?
That's right. Yeah, no, it's my website is sentinel.com, so you can reach out there. I always tell people, like, I didn't expect it to go this route with social media being the way that a lot of people like to reach out. So reach out that way too if you'd like, and I'll take a look if you have some issues or if you just have, like you had yourself, right?
Exactly. They're everywhere.
They're
Uh,
everywhere. I always like a target rich environment, so no problem. Give me a call.
Well, sometimes it's really hard to get a good picture of something or a good video of something because some of these
Yeah,
things are very tiny. And
and
so,
and
like,
fast.
yeah, sending you a blurry picture of something sometimes isn't that helpful. And you're like, I don't know what the hell that is. Give me a
I just
video.
want to be sure. It's like the worst I could possibly do is say like get a little bit of info, be like, I think it might be this, and then it not be, right? So yeah.
Yeah,
But I become an expert, so try me, challenge me with the blurry photos. We'll see
yeah.
what we can do.
Is it easier if you have a video because you can kind of see how they're moving?
Yeah, I would say so. If you can like train a video with the right focus for even like 5 seconds, it's usually enough.
Okay. So we know you do. We know how to find you. Great videos that you have. I feel like this social media thing really changed during COVID. People kind
Yeah.
of had to retreat to their homes and find other ways to connect. And that's definitely where Clubhouse took off. That's where we met each other. I think LinkedIn is one of the cannabis friendly spaces where there's a lot of serious people. It's not just smoking buds and rolling blunts, it's people who are scientists who are really enthusiastic and experts in multiple different fields in the cannabis space, which is kind of my goal here with MyFlora Fridays is to have experts come on, talk about their various wheelhouses and get information from people who really have a lot of expertise and a lot of wisdom in their spaces. And so I really appreciate your time and your wisdom and all that you do for the cannabis community with your research and just your general willingness to help out. So thank you so much. Is there anything you want to add? Is there anything that we didn't talk about that you really wanted to?
We actually got to talk about pretty much everything I wanted to talk about. I might have had a few slides in preparation, but we talked about the ones that were most important to me, and I think that were most valuable to people. So the last thing I want to say is that I would love it if people would take this as inspiration to just look at pests a little bit differently. Don't look at them as like an ultimate evil, because they're not. And don't look at them as something that is like incapable of changing or not being what you think that they're going to be. Certainly, the videos and the media and the information I put out is meant to articulate their capabilities as much as possible, but emergent things happen. Your situation is not always the same, and just have a healthy respect for not only the plants that you grow and their abilities, but the things that are trying to eat them too. So that way we can be just more effective as a community. And I think that would do a lot for us because it's a healthy respect for nature ultimately.
Mm-hmm. Thank you. I appreciate, again, all your time. We like apples, we like cannabis, but so do the bugs.
Exactly.
And yeah, so it's been a really nice conversation. Thanks as always, Matt. I appreciate you.
Well, I look forward to our mutual success. Thanks so much. Have a good
Thank
one.
you. All right, I'll talk to you later. Bye.